All right. Yes, so this is the third workshop. I'll just pop that open here. We're going to talk about running a complete workflow, a bulk RNA-seq differential expression workflow. And my kind of thoughts about this were that kind of familiar to many of us. It's not super complicated. It's kind of a well-worn path. We know how it connects to an interactive analysis in Bioconductor through the DE-seq package. And also, it's quite difficult if you're just running on your laptop. One part of it, the FASTQ files to count matrix is relatively complicated or requires a different set of skills from the interactive analysis. So it seems kind of convenient to think about how that might work in the cloud. So it seemed like a good topic. And Terra actually has been developed the workspace that we'll use. It's been great to be able to tap into it for this course. So let's see, what do we got? Going to be looking at this workflow. So it's bulk RNA-seq differential expression analysis. The idea is you've sequenced a bunch of samples, RNA-seq sequencing of a bunch of samples, and you have FASTQ files. The FASTQ files need to be aligned to a reference transcriptome, and they're these so-called ultra-fast aligners, Salmon and Callisto and so on, where ultra-fast means that they take, say, 10 or 15 minutes per FASTQ file to do the alignments. And what happens is that the alignment occurs, and then you have outputs from the alignment, and those outputs can be assembled into a matrix of counts where you have genes as rows and samples as columns, and the individual entries in the matrix are the number of reads that align to that particular gene and that particular sample. And that kind of count matrix works into this DE-seq2 package, which has a totally excellent vignette describing how to do bulk RNA-seq differential expression analysis. So that's kind of the big overview. Of course, we're going to be using AnVIL. The Salmon website and the excellent DE-seq2 vignette are excellent reference materials. In terms of the workflows, they're written in something called the workflow description language, and this is a reference to a definition of that. And you run workflows locally, like on your laptop, maybe as you're developing the workflow using some Java software called Cromwell. And the workflows make use of so-called Docker containers to containerize the software environment in which the different tasks in the workflow are going to be run. So there's some key resources. Then just kind of catching up for those of you who may have missed a session or two. In the previous workshops, there are links to videos that can be not so terrible to watch. Each one's about an hour that you can review. We've learned how to log into the AnVIL, how to navigate workspaces, the way that you associate a billing account with the computing tasks that you're going to do. And we've explored Jupyter notebooks as well as RStudio for interactive analysis. Last week we focused exclusively on RStudio, and this week we're going to be using the notebook, so we'll kind of get a flavor for each of those interactive environments. And then there are also these workflows for workflows, find the right place to double click, workflows for large scale data processing, which is what we're focusing on today, FASTQ files to count matrices using Salmon. We've also explored the compute environment of the interactive environment, the runtime and persistent disk. Last week we saw data elements in the workspace, and we were also exposed to Google Buckets, which is where we can save large data results. And actually, the workflows will write out the results to the buckets. And then we also saw the AnVIL-Bioconductor package for interacting with workspace components, and that'll come up again. There are a bunch of questions that came up in the chat, which I'd encourage you to use as we're going along now, that I've tried to capture in the frequently asked questions section of the last notes. I updated them after the fact. Liz, who's an expert and is on this call, pointed us to the Broad Methods repository and also the WDL Puzzles workspace. She is also the author of the HCA Optimist Pipeline workspace, one of the authors that we worked through last week. So it's great for Liz to be here to keep us on the straight and narrow with these things. Levi asked about when you did AV Workspace, how it knew this information. And that information is actually encoded in system variables in the runtimes that we're using. AV Workspace consulted those system variables and returned that information by default. And Levi also pointed out that if you try to copy a very large or larger object, you got a warning about this, and then actually it failed with an error. And that was a bug that needs to be fixed in the underlying runtimes, and it'll be fixed the next time those runtimes are updated. So actually very useful stuff going on in the FAQ and opportunities to learn stuff, from presenting the workshop, learn about problems, and great that you're flexible enough to roll with the punches. All right, so that's kind of catching up. Were there any questions briefly? Just type up if you want to ask. All right, so what I wanted to do this time was I wanted to run a workflow, and I also wanted to do some pretty update our Jupyter Notebook runtime. And these are actually time consuming, so we actually have to jump right in and not spend too much time getting things started. Otherwise, we won't get finished before the workshop ends. So what I'm going to do is I've gone to AnVIL Terra Bio, and I hope that you're following along, right? And I'm going to sign in with Google, and I'm going to use the account, my Google account that's associated with AnVIL. So if you've been to the workshops before, this should be familiar territory. And then I'll go to Workspaces. And you should see this Bioconductor Workflow DE-Seq2 workspace. And if you don't, just shoot Kayla a message with the name of the AnVIL account that you use to log in and she'll add you to the pop-up group that gives access to this. You can see it was developed a little while ago. And what I wanted to do was I wanted to make a clone of it. Actually, I've got a clone from earlier. It's kind of like it's in the oven, it's already been cooked. But I wanted to make a clone, and I want you to make a clone of this DE-Seq2 workflow. And name it something unique. So I'll call this MT Morgan Live. And then the Billin project should be, you should have access to Deep Pilot Bioconductor May 10. And again, if you don't have that information, then Kayla can help you set that up. So clone the workspace. So I'm hoping you're following along here. So we've got a cloned workspace. Are there any questions? Andrew indicated that he wasn't going to be able to join us today. He had a scheduling conflict, so I'm relying on other people out there to chime in with problems that they're having. So I hope we've all got our own clone of the workspace now. And I wanted to get our runtime going and then come back to it. So I'm going to go up to this cloud environment, and when I click on it, you'll see a box that's slightly different when you click on it. When I click on it, that's slightly different. But I want to use this default. I want to use the Jupyter Notebook environment. So I don't want to use RStudio this time. I want to use the Jupyter Notebook environment. It should be the default environment. And then I think we'll just stick with the standard CPUs and memory and persistent disk. And I think that you guys will have a create button here. So you should press create, and I'm going to press next, but we should end up in the same place. It'll say, it should say up here in the corner, cloud environment creating. So we should be creating our cloud environment. It says 6 cents per hour. I'll say that last week our workshop spent $1.55 for all of us. So this week I'm really hoping that we can really boost the cost and maybe get up to like $10 for the day. I don't know. We'll see how it goes. So that's one thing I wanted to do. But kind of while that's in the oven, I'm going to just duplicate my browser tab. So now I've got over here, I'm waiting for Terra to load and being impatient. So I'll just like reload it again, which seemed to work. So now I've got, this is where I'm waiting for my cloud environment to start. And over here, I wanted to start our workflow. So I'm going to go to this workflow tab, right? And I'm going to click on it. And this is the workflow that we're going to run there. You could have multiple workflows in a workspace. This is the only workflow in this workspace. It's stored on Dockstore and GitHub, but we want to really just jump right in as quickly as possible to get the workflow running. So I'm going to, you looked up at the clock and realizing that time's going by pretty quickly. So I'm just going to walk through this very briefly. We're just going to click some buttons kind of without any deep thought about this, just so that we get the workflow running. So the first, it says run workflows with inputs defined by data table. So it says we're going to look at the data tables, and in particular this participant set data table, and we're going to use it for inputs to the workflow. So here's the data table that has a bunch of participants that actually references this participants table. We've got a bunch of FASTQ files here that are available. And it says, where are we at? It says run the workflows with information defined in this participant set table. And then the next step is select data. And the idea of this participant set is that you could like maybe slice and dice your overall data into like say different populations or different protocols, or maybe a subset for developing versus the entire analysis. This only has one set. It actually consists of all of the entities. So these are the 16 participants that are defined in this set, and they're the ones that we're going to use. So I'm going to click OK, and I'm hoping that you're following along. So again, we had run workflows with inputs defined by participant set, select data, chose the one set, and clicked OK. So now that tells us where the data is coming from. And then down below, this is the script that we're going to run that does two things that we'll get back to. It creates an index for performing the alignment, and it actually does the quantification of each of the FASTQ files to the reference index. And to run the script, we need to associate inputs from the data that we've defined with arguments to the script. And the table here has the values predefined. So we'll come back to this. I need to go to outputs. So inputs is good. Move to outputs. So when the script runs, it produces outputs, and it's going to store the outputs in the Google bucket associated with our workspace. And it says, okay, I'm going to store the outputs in the Google bucket, and how do you want to reference them in the results? And it says, well, I'm going to take the participant set table, and I'm going to add some information, a column called file, and an array of columns files that represent the quantification outputs. And so what I'm going to do is actually I'm going to click this Use Default button, and that says what the person script said you should do by default. It'll add a column to this tables participant set called transcriptome index, and it'll also add a column to the participant set called font output. And this says it's referring to where the reference the outputs will be added. And it's actually the actual outputs will be in the Google bucket, and what will be added to this participant set file are links to the Google bucket. So we can come back and talk about that a little bit more. What I need to do is then press this Save button, and now that I've got everything saved, I press this Run Analysis button. So I see Levi scratching his head and stretching and stuff, so I can't believe he's following along, which is too bad because now we're just going to spend $1.05 on this workshop as well. But what I really want to do is I want to run this analysis. So I hope you can catch up. I know Levi's a pretty quick study, so maybe he's already caught up. So then I just
my head was just itchy.
And it says you're going to do a bunch of analyses. It's going to be in this part of the Google Cloud, and do I really want to do it? And it's worth being sure that you really want to do this. And I'll click Launch. And then what I've actually done is I've submitted my workflow to the Google Cloud, and it's started, well, it's queued up. So it knows it's going to run this script with the data and producing the outputs that I indicated and that I hope that you are. And now it's marshaling the resources to run the workflow. So we've put that in the oven. Very briefly, did people follow along? Is that okay? Sorry, Levi, for shaming you publicly like that. It's not a very professional thing to have done.
Yeah, I have no problems with it.
Excellent. Great. Wonderfully. I get a little
mine's running too, and my head's not itchy anymore.
Excellent. Yeah, multitasking. This is great. So yeah, so what I'm going to do is I'm going to continue to multitask. So we've got our computers going, but now I wanted to return to the notebooks. I guess I'd open this other tab in my browser. Oh, look, looks like mine has started to run while we're waiting. I'm going to return to this other tab in my browser, and you can see that I've got my cloud environment and it's running. And we could actually go to one of these notebooks. So I went to the Notebooks tab. Here's my pet peeve. I've probably said this a bazillion times already. I always want these to be sorted alphabetically, and every time you navigate away from this notebook page, it reverts to sorting them by last modified date. And usually you'll modify the last thing that you did. It might make sense in some circumstances. It's always wrong for me, so I'd like it to stick at alphabetical. So I just wanted to open this overview notebook. So you remember that we have these different runtimes. We have an RStudio runtime, but this time we launched the Jupyter Notebook environment, right? So now we've got this notebook, and the notebook is, kind of like a markdown in some ways. It's got different types of cells. Right now we're in preview or read-only mode, and I'm going to click this Edit button. And what it'll do, it'll actually copy. The notebook is actually in the Google bucket associated with my workspace, and it's copied it over to my runtime, to the persistent disk in my runtime. And then it's opened it again, but now these cells can be fully evaluated. And we could go through it. And the first thing that we need to do, and this is also a time-limiting step, it'll be amazing if we actually end up getting through just the setup step of this. But so we actually need to install a bunch of packages on our runtime. I'm really hoping that in the next iteration of the AnVIL, this step will be really fast, orders of magnitude faster. But actually, it takes quite a while. And I don't want to do it in the Jupyter Notebook, because what happens in the Jupyter Notebook is that if I were to just press the carriage return at this location here, it would evaluate these commands and everything would be great. It would indicate that it was running with the little asterisks here, And then all of the time that packages are being installed, it would just sit there and be completely boring, and you'd lose confidence that it was actually running. So I wanted to see the packages being installed, just like, because it'll make the time go by faster. And so what I'm going to do is I'm going to copy this stuff, just, drag over and Control C or whatever to copy this stuff. And then I'm going to go up to this thing, this little icon in the top that says Open Terminal. So this is like my runtime, and then this is like a little window, a terminal window on my runtime. And now what I've got is a terminal, Jupyter user app, blah, blah, blah. And here I can do all of my favorite Linux commands. And in particular, I can start R like this. And then I can paste the magic commands into the terminal window. And now what it's going to do is we'll just see. The first one was it made sure that the BIOC manager package was installed. And then this line, BIOC manager install ask equals false, it updates all of the existing packages on the Docker image. And in many ways, this is the worst time of the release cycle to do this. We're right before the next release, and the Docker image was released right after about 6 months ago. So we actually have to update about 6 months worth of packages. And you can see we're doing the usual sort of things. We're downloading a bunch of files, a bunch of packages. And actually, this is cloud-based transfer, cloud to cloud. So the downloads are actually going really quickly. And now that we've downloaded the packages, it's starting to install them, going along here, installing the packages. And it'll take a little while. So we've got multiple ovens going on here. We've got our workflow running, and we've also got our packages being installed. The workflow is running in one part of the Google Cloud. The packages are being installed in another part. And I'll just pause and have people follow along.
There's one question in the chat about someone had an issue pasting into the terminal from the notebook.
Was there, let's see if I can find this chat here.
Sorry, then the terminal actually for some reason died, and I'm trying to get back into it, and nothing is not loading.
I see.
Unfortunately, so it could be something on my end.
Does the terminal does it just show a black screen, like nothing at all?
Previously, I started R and then tried to paste, but maybe it just decided it didn't want to work today.
Yeah, it'll be difficult, a little bit difficult to debug, but actually one of the surprising things that I found was that when I reconnected to the terminal, it was actually at the R prompt, and it was just waiting for me to press the carriage return. I'm not sure whether they're actually just pressing the carriage return brought it back to life.
All right, I'll try it again.
Yeah.
Martin, last time Sorry, I beg your pardon. Martin, last time when we were using Anvil, we were able to install binary versions of the packages super quick and nice, and that was a great feature. Why are we having to go back to the old school? Yeah,
because it's a technical issue, and so the binaries are meant to be a feature. They're just irresistible because they're so fast, right? So the binaries are meant to be a feature that'll be incorporated in the next release, but they're currently only at the RStudio image and the Jupyter image use a different base version of Ubuntu. So the Jupyter image uses 1804, the RStudio image uses 2004, and we're only building binaries currently for the 2004 image. So only the RStudio image can install, but there are only binaries to install for the RStudio image. And what will happen at the next release is either one of two things. We're hoping that we'll have a common Ubuntu 2004 image, and then we have only one set of binaries to build, or we'll build binaries for both images, and then they'll be fast installed. And this will be much less painful.
Yeah, I can't copy and paste, unfortunately.
So what you could do, is it, I think is it Elizabeth or?
Yes, it's Elizabeth.
Yeah. So I think what you could do is just in your Jupyter notebook, you can just press the
Yeah, I can run
button
it there.
here,
All right.
or,
I just wanted to make sure it's okay
right,
to just
yeah.
run it in Jupyter. Okay.
Yeah, it'll produce a little output, and then it'll be completely boring for 20 minutes or so, and you'll think that the world has ended or time has stopped or something. But so yeah, you can.
It's giving me some output, so I can see that it's running.
Yeah. Okay. All right. So that's great. So I think we should go back and look at our. Were there other things? So that's kind of like the stuff we have to do right at the front to just get things, have any hope of finishing things during the workshop. Were there other questions that people wanted to talk about? Yeah. Okay. So I'm going to go back and look at this workflow here. And it says it's running. I think what I wanted to do was I'm a little bit shy of navigating away from this, but I'm just going to duplicate this tab one more time. And I had a bad experience when I was preparing this stuff, and I couldn't get back to this display, so I'm just going to keep it there and see how it goes. So I'm going to go back to the job history. It's going to go back to the workflows and open this workflow again, and then just walk through the workflow just a little bit, like a second time, but a little bit more relaxed, so we can get a better feel of what it is that we're doing. So first of all, it says the workflow comes from a GitHub repository. And this is a repository that Kayla has been working on. So we could probably open it. And it says, so that it's a GitHub repository that comes through DocStore. You can go back to the repository. And actually, Kayla has, just as convention, has put her workflow in this location. She's organized things as an R package, but she could have stored her workflow, at the top level of a GitHub repository. It doesn't really matter, but it turns out to be beneficial to store it as an R package. And if you know about R packages, you'll just notice in this vignettes folder that those Jupyter notebooks turn out to be R Markdown documents. So that'll be kind of fun a couple of weeks from now. And then in this sort of traditional location, the inst location in an R package, there's a folder called workflow that Kayla invented, but it's a very useful convention that we'll follow in AnVIL and R going forward. And in the workflow, she's defined the workflow in this language called WDL, Workflow Description Language. And the workflow consists of things called tasks and the workflow itself. So this first task says it's called Salmon Index. It takes a couple of inputs, a FASTA file and a name, and then it runs some commands, and it runs the commands on a particular runtime on a Docker image, and then it produces some outputs. So that's like the first task. And if you know anything about salmon, what this is doing is it's creating an index of the FASTA file so that the alignments can perform really quickly. And you create an index once for each transcriptome file. And in some ways, since we're all using the same transcriptome, we could have rearranged the workspace so that the index was already part of the Google bucket in the workspace, and we could have just copied it. We could have used that pre-computed index, but it's a little bit more fun to do the whole analysis here. And it might be the case that you have a FASTA file that not to be the organism for which we're working on. So we've got a task, consists of a command, and these things are just like shell commands, right? But since they're shell commands, they could be any command that the shell does. So you could like launch an R script or something like that in the command. So they're shell commands. And then this runtime is the computational environment that the shell commands are running on. And you can see that we're using a Docker image. And a Docker image is a collection of software produced in a reproducible manner. And this Docker image comes from the lab that produced the salmon software, so that it's like kind of definitive, right? It's what the pros say, the people who are most knowledgeable of the software say, if you wanted to use Docker to run this software, you should use this Docker image. So we're saying they know what they're doing. We don't need to reinvent anything. And we've pinned it to a particular version of their software so that no matter, which is a blessing and a curse. So this is a fully reproducible workflow. We'll always end up with the same image. But of course, if they fix bugs, we don't get the bug fixes. We get the same bugs as we always did, but it's reproducible. Terra wrote this. It's just a plain old text file. And she had to figure out what the commands were to create the salmon index. You visit the salmon page, it has documentation, it describes how to do that. But in some ways, Terra, the bioinformatician, gained that expertise once, and then now all of us are benefiting from that. We don't have to discover how salmon creates an index, and so on.
Can I interrupt you just for a second? Can I interrupt you just for a second? So the task says salmon index, and underneath it says file, transcriptome FASTA, and string transcriptome index name. So this is a task that's expecting to receive two components?
Yes, two inputs, right.
All right, and does the order of the inputs matter?
No, I don't think so. This is great to think about that, but I think it's basically pattern matching. So it's expecting a file that has the name transcriptome _ FASTA. But Liz Kiernan knows better than me.
Well, I think too later in the workflow stuff, when you define these, I think, like you said, I think it's just going to pattern match. I don't think it necessarily depends on it.
Right.
Yeah.
Yeah.
I believe I believe that's correct too.
Yeah. Okay, great. Yeah. So it says it's not totally transparent, but it says the input name transcriptome FASTA is given to the value of transcriptome FASTA in this workflow. So, yeah. And actually, these things have been defined when we created the workflow. We'll come back and think about that. But, yeah, great.
Martin, if we wanted to run this on our own data, would it just be a matter of replacing the participant and participant set files?
Yes. So you could, I'm not sure, I think this is probably, so you could go back to your workspace, and the idea, if you're going to run your own data, you could replace participant and participant set, but you could also create your own table called, Levi's experiment that had, it doesn't have to have any information other than the links to the two FASTQ files. And actually the links to the two FASTQ files these guys turn out to be files that are in the Google bucket of the workspace that we cloned. So like when Kayla was creating this workspace, she placed those files onto the Google bucket of the workspace. So, and she actually did it programmatically, and the way that she did it programmatically is described in one of these notebooks, probably organizing data. So she probably talks about, like, this is kind of a special case, right? Like I think she queried the SRA for the short read archive for the particular files. She downloaded them and then copied them to the bucket. That's how she got her FASTQ files there. But you could imagine that what you did was you went to this Files tab and you clicked on this plus button and uploaded the files to a location on the Google bucket associated with your workspace. So it's like, yes, you'd
are there any good shortcuts for making these files?
Well, so like, I mean, it really depends on where the data is coming from, right? And probably at some level, this condition and type over here, these two columns, are the experimental design. And so somewhere in your R or Excel spreadsheet or whatever, you've got a table that says, these are my participant IDs. They were subject to these different components of the experimental design, and the FASTQ files associated with those guys were there. So maybe it's like an Excel spreadsheet that you export to a CSV file and upload the TSV file to tables. Or maybe you've done something programmatically in R where you've done all of those steps to create the table.
But then the file itself isn't just these file names, like it has the GS links, right? So it would just be like a search and replace or something to add to prepend those GS / to it. Yeah.
So like it's a little bit hard to see here, but the GS says the GS // and then the first number FC-78B blah blah blah. That's like the bucket identifier where it's stored. And that's determined by the workspace that you're going to store them in. But then the rest, data DRO16125, DRO16125_1, that's like Kayla's bioinformatics convention, or the convention that you and I as investigators are using to, as individual investigators, just the way that we're going to organize the data. I'll mention also that the steps of creating this table and adding it, and also of uploading the files, can be. You can write a script. You can use the AnVIL package to do that, to do both of those operations. And actually, we saw last week that you can run the AnVIL file when you're outside of the AnVIL package when you're outside of the AnVIL environment. So if it were me, and I had a bunch of FASTQ files that I wanted to move up to a Google bucket, I'd actually run the commands using gsutil_cp from my local laptop or my local workstation, referencing the local FASTQ files and telling it to copy them up to the Google bucket associated with my workspace. So I've done that programmatically. That's great. So index That's in many ways, Levi, that is a great question, and the idea of analyzing your own data is, of course, what you want to do. You don't want to always analyze, figure out how to make it extensible that way. So the first task created the index, the transcriptome index, and then the next task takes the two FASTQ files from a single sample, and it performs the quantification. And again, this magic command here is from studying the salmon documentation, and it says do this magic command, but use the transcriptome index that you created in the previous task and the FASTQ files that were on the Google bucket, and perform the alignment. And we've got a runtime here, the same runtime as before. I should say too, this came up last week, the runtime has additional flags, so you could specify the amount of CPUs that you wanted to request, and the amount of memory and the size of the disk and other features of this runtime. We've just opted for the default, which is probably good enough for the task at hand. It's maybe a little inefficient, a little over allocated, but then we didn't have to worry about optimizing it. It's not going to be an arm and a leg anyway. But we could be, in particular, we could have asked maybe this indexing step. Maybe it takes quite a bit of memory, and we could have asked for a very large runtime in terms of memory at this stage here, and then use the smaller runtime for these more efficient, more space-efficient tasks. So that's the quantification task. And then these guys are assembled into a workflow. Ludwig, did you want to say something, or just? Okay, so the workflow says these are the inputs, and if we were to look back at when we launched the workflow, we had to provide this information, the inputs to the workflow. We had to provide a file that was transcriptome FASTA, and we had to provide an array of files. So array of files in R, this would be a vector, a column of FASTQs, and another column of FASTQs, the first and the second end. And we also needed to provide a string that indexed the transcriptome index name. I think maybe I can go back to here. So you can see the variable is called FASTQs1, and it comes from the participants table, the FASTQ1 column of the participants table. And if we, used a different table, then we could have changed those entries here and just mapped the variable that the workflow needs to whatever the variable was in our data set. You'll also notice the transcriptome FASTA file. It's a file, and we've just entered the location, the cloud location of this transcriptome FASTA file. And Kayla, when she set up the workspace, copied the transcriptome FASTA file to this location in the workspace bucket. So it's just a reference to the Google bucket of the workspace, of the parent workspace. And then this AnVIL index was just a name that Kayla chose. She could have called it whatever she liked. She was following the convention in the salmon documentation. So AnVIL index. So like these variables come from the workflow inputs, and the mapping to values in our workspace are done by these types of operations. I think that this gives you so these types of operations. So that's the workflow so this is the workflow. It says, I need these inputs, and the AnVIL has provided a way to map the inputs needed by the workflow to the data that's in the workspace. Is that good? All right. So the workflow says, take some inputs, then invoke the task, the salmon index task. We saw the mapping between the inputs needed by the salmon index and the inputs provided to the workflow. And then after performing the index, it then calls salmon quant, again with inputs. And then it's just interesting here, this salmon index dot transcriptome index, that says consult the salmon index task and look for an output called transcriptome index, and use that as the transcriptome index of the salmon quant task. So you look at salmon, the salmon index task, it has an output called transcriptome index, which is a file name that was created by these commands. So this is the output of the salmon index task, and we use the salmon index output as inputs to this next step in the workflow. And then, so this calls salmon quant, but it actually calls salmon quant on just a single file. And this scatter is kind of like a for loop. It says, I guess, reading from the inside out, it says, create a data structure that zips together we have these two vectors of FASTQ files, and it creates pairs of them. The first FASTQ1 and FASTQ2, the second FASTQ1 and FASTQ2, the third. So it zips those things together, and then it iterates over that zipped together list. And the FASTQs then has a left part, which is the FASTQs1, and a right part, the FASTQs2, the corresponding element. So we're going to iterate over each of our samples. Okay, so we've got like this zipping together, deconstructing the zipped parts, iterating over them. And then the skater is a little bit scatter, is a little bit magical. It says, oh, I see you've got a vector of 16 pairs of FASTQ files. So I'm going to take each element of the vector and I'm going to distribute it to a different compute node. So I'm actually going to call salmon quant in parallel using 16 compute nodes instead of one. So that instead of executing the quantification sequentially and taking 16 units of time, I'll parallelize them and take 1 unit of time. So we'll do the quantifications in parallel. And then you can see that the output is a tar file, quant output. And the quant output, it's an output of the workflow, and it'll actually be stored on the Google bucket of our workspace. Sound good?
Sounds good to me, Martin. Thanks.
Good. Yeah. Okay. Great. Yeah. All right. So now, oh, cool. So now I'm going to go over to the oven and check on my workflow to see what's happened to it. And it says succeed. That means that workflow that we just walked through while you and I were trying to understand it, the computer was saying, okay, I'm doing what you told me to do, and I'm going to execute it. And I think what I want to do is these links, I want to look at this job manager link. Did you guys have success with your workflows? Are they completed as well?
Yeah.
Completed.
Yes.
Excellent. So I think that we've probably managed to spend a little bit of money on this workshop, so that's great. So you can see list view, salmon index, salmon quant, ran it when I started it, how long it took for each of the steps, 17 minutes for the quantifications and so on. It says 0 minutes for the salmon index stage. That doesn't seem quite right. We could review the inputs and the outputs. This is actually like the expanded version of the vector from the column of that table that the FASTQ1s and FASTQ2s were at. These are the outputs where they've been written, and you'll notice they've actually been written to the Google bucket associated with our personal workspace. So we've got the transcriptome index and the quantification on the Google bucket of our workspace. We'll hopefully be able to see that. And then this timing diagram is totally great. Oh, that's really clever of it. Totally clever of it. So, so this shows the different stages. This first step is the index quantification, the creation of the index, and then all of these things that started at the same time, these 16 things, those are so this is creating the index, and then this is salmon underscore index, and then each of these guys are those parallel jobs doing the alignments, the salmon quant for the 16 different samples. You can see that some samples, the different colors represent different stages. Some samples processed quickly. The timeline along the bottom starts at like 22 minutes after the hour and goes until 33 and a half minutes after the hour. So this quantification took 11 minutes, whereas this sample, I guess it was larger. It took 40 minutes. It took almost I guess it was a 17-minute one. But they were run in parallel, so we just spent the total execution time took as long as was necessary for the longest task. That's pretty cool, right? Like it just did the task in parallel without any effort on our part.
Martin,
yeah.
If I may ask, if I recall correctly, the virtual machine that we allocated had only one CPU, or how is it possible that it runs all these in parallel?
Yeah, well, actually, there's like the tasks are actually run on their own, like independently of our runtime. Like our runtime, like the Jupyter Notebook and the infrastructure that's behind that is independent of the infrastructure that the workflow is running on. Kind of like the beauty of the cloud, right? Like the Google gave us some compute power for our runtime, but then it also allocated separate compute power for each of these steps. And actually in this scatter, the workflow engine said, I don't want one compute node, I want 16 compute, blocks of compute nodes. So we actually got 16 of whatever the runtime was we specified here.
But this means that a mistake in the workflow language script may put you into trouble with your billing thing, right?
Yes. So here's where you could really, you could make a bad mistake. Like, I don't know, like if you somehow phrase this as, like do it for each read in a FASTQ file, like you got it transposed or something like that, and then like 100 million reads. Probably the Google Cloud would say, okay, I've got 100 million compute nodes for you. Here you go. And then you'd really spend some money. So yes, things could go terribly wrong.
Mm-hmm. Okay.
So where are these files now? How can I get them into my notebook? Yeah.
Well, right. So we're at exactly 59 minutes. So we're totally out of time here. But if you go back to our notebooks, you can see notebook D, managing workflow output. So I'm guessing actually we'll just carry on just a little bit, it looks like, oh, darn, it asked for something, and that means it didn't install. It got only partway through the installation, so. Okay, maybe that's fine. Maybe that's good enough. And anyway, so we might have to come back to that just a little bit. But in this managing workflow output, we could launch our Jupyter notebook and load the AnVIL package. So I entered that cell and pressed the carriage return. I'm just going to see what we got here. Great. Actually, this is the development version of the AnVIL package. So I think that the script, even though it ran into problems, I'm not sure. Maybe there are problems here. Yeah. Maybe you guys didn't have problems. Maybe your scripts ran all the way to completion, your installations.
So mine ran to completion, but it left it dangling, Martin, with the AnVIL install, the same package that yours was still on the clipboard there. So I have to just install that separately. Yeah. I don't have it.
Yeah, well, just press the carriage return and it'll install that, and it won't take very long. Yeah, that's great. Exactly. Yeah. So actually, what this magic command does, what this magic command does is it finds the workflow files that were created by the workflow. And actually, if we go to data and we look in so this is our Google bucket. And this identifier is actually the identifier of that last workflow that we ran. And when you look inside it, you can see that there are files that have been artifacts of the build process. So these are artifacts of the workflow process. And as you drill down in them, you can see eventually these DRR blah blah blah. This is actually the output that we're interested in. So the workflow wrote the outputs to the Google bucket, as well as a whole bunch of diagnostic stuff. And there's some sort of fancy path in the Google bucket that leads to the files that we're interested in. And so if we had like about 4 times as much time available as allocated, we could walk through this. This says, grab the file, the path from the Google bucket. This says, copy them from the Google bucket to the local working directory. This says, untar the files. And then we've got the outputs of Salmon on our local runtime. So the workflow outputs have been transferred to our local runtime. And then in the final notebook that we'll mention, we take the files that we've just localized, and we use the metadata about the experiment that's in our data table, and we create this object that we can use in DESeq, in a standard DESeq2 workflow, using this TX import function. So like the 2 notebooks that we, of course, didn't get a chance to work through, bring the files from the Google bucket where they're produced by the workflow to our local runtime, and then incorporate them into the data structure that we need to run the workflow. So we're totally out of time, of course. I'll mention that the interactive analysis section here takes us through those steps. And if we were to just jump to what we've accomplished, we've seen how you can run a workflow. We've explored the workflow components, and we've really just touched on interactive analysis. so pretty action-packed session. I'll stop here, but I'm happy to stick around for questions. Sorry that it's so packed.
Thanks, Martin. That was great.
Yeah, thanks, Martin. That was excellent. I was wondering if maybe Kayla could comment about her experience. You did a great job organizing everything and setting everything up, so even I could understand what was going on in these parts, which is no mean feat. But like for you, Kayla, like is this a lot more work than spinning up a regular instance somewhere in the cloud and running your normal workflow with your bash scripts? And like what big advantage do you see if you're not?
Yeah, I guess so one of the advantages, I was working on a different workflow, actually using like attack seek data and things like that, and I was running into really, really large data sets. So I liked the availability for Anvil to be able to store the files on the Google bucket and kind of have everything setting it up. You have a lot of moving pieces, like I have the scripts on GitHub, and then I have to read them into DocStore, and then from DocStore I have to read them into Anvil. But once you have the workflow running and everything's just in one location, I think Anvil has it really nicely set up that you can just pick the inputs, set the inputs, set the outputs, run the analysis, and then you can kind of interactively get an analysis together in a notebook. So I think from a user's perspective, I think it's nice to have everything in one location. But if you're developing, it can be a little time intensive trying to get all the moving pieces together. But once you get a nice working workflow, like this DE-Seek one, I think went really smoothly. I think it saves a lot of time, and everything's just in one place.
Just a follow-up question there, Kayla. If you develop this workflow using the local installation of Cromwell, are there any changes you have to make to run it on Terra, or it's exactly the same?
It's pretty much exactly the same. So I kind of start locally, at least for this workflow. I had started with the attack seek locally with a small data set that I could actually have on my local computer before I started running out of space and such. But I can grab, like for the DE-Seek, I was able to download the data locally, and I could that's kind of where I did a lot of the debugging in the workflow script, is just locally running it with Cromwell. And then once I knew that the script was somewhat runnable, then I would upload it into DocStore, and then I would find little bugs in the upload process. And then I would get into Anvil, and then I'd maybe find little bugs in Anvil, but it definitely helps being able to work on it locally to get the big bugs out of the way, and then you kind of find little ones along the way that are much easier to fix.
Thank you. That sounds great. Thanks. And thanks for all your work on it. It's very much appreciated. So then do you envisage like running more like routine analysis in this kind of environment, Kayla, rather than... Sorry, I'm getting visitors. Hold on.
I mean, I honestly, this was just kind of something that Martin brought to me to say, hey, you want to work on this? And I worked on it. So I don't know the big picture as to, obviously there's probably people on the call that can see in a bigger picture how this could be usable, whether it's in a course or in their own work and things like that. I'm open, especially with this call, if people see ways that the workflow can be improved, that instead of just, for me, it was, I'm going to write this script so that it works and that it runs, but I don't necessarily see down the road the big picture as to how often it'll be used and things like that. So I guess that's kind of everybody else on the call that might actually use it on a day-to-day basis or something like that, how it might be useful and things like that.
One more question. If you add one more pair of FASTQ files to your participant set and click run on the workflow again, will it run all 17 of them or just the new one?
I believe you can set in the inputs where we had the participants table, we ran it on all 16 entries. You can pick a specific sample pair. So when I was working on the attack-seek stuff, to debug, I kind of was just running it on one pair because it takes a really long time to run. So I knew if I could get it running on one pair of samples, then I kind of scale it up to I think I was working with 5 or 6. But you can just pick a single pair if you want to just run it on that.
But it's not checking the presence of the outputs before it runs. It will, if you choose all 17, it will just run all 17 even if the output files are already present.
Yes, because you're going to get a different job ID. So then you'd have it's an unnecessary run, obviously, but you're going to have a new job ID. So then it's not going to know that you had done a previous job. It's not aware of that. So you'd be running the whole analysis over again. Yeah.
Actually, I'm not exactly sure whether that's entirely true, and I'll tell you. So I see this. I'll clarify with see if I can't get a.
Oh, that's yeah.
But use call caching, and the reason that I was really surprised at how quickly this step took. Like it says, it took 18 seconds, but actually the quantification can take tens of minutes, and I think that it actually. Like 10 minutes. I think it actually recognized that this step was the same as something it had done before for me, maybe when I was demoing things. So at least this step, it recognized as something it didn't have to repeat. So it wouldn't be entirely surprising that maybe some of these other steps could also be cached. Maybe there's something that could be explored there. See, we've lost Liz, our local expert, but I don't know if anyone else has any experience there.
If I may ask, I have the perception that one has to do a fair amount of point and click to allocate the resources and get everything configured. Is there any way to do the same kind of job of configuring stuff on command line, like a command line client for AnVIL or something like that? Because also for reproducibility, that could be great to have everything written, right?
Yeah, there are two things. So you can see that you can download or upload JSON. So you could have a text representation, but also in the AnVIL package, you can configure the workflow. There's a AV workflow _ something or other that allows you to configure the workflow. You can do everything, but actually click the run button programmatically.
Mm-hmm. Okay. Great.
All right, thanks. So the recording should be available later today or tomorrow. Next week, Vince is going to talk about Vince Carey, so a different presenter, someone with a more level, calm voice. We'll talk about orchestrating single cell analysis, so single cell analysis in the context of AnVIL. And the week after, Levi is going to talk about teaching. All right, great. Thanks so much for showing up and following along. See ya.
Thank you, Martin.
Thank you, Martin. Thank you, Kala.